Hello, everyone, and thank you for joining us for today's webinar, Peptide-Based Therapeutics, Developing and Scaling Through Innovative Technologies. I'm Heather Masaccio, your host from Outsourced Pharma. With me today, I have Adrian Amador, PhD Director of Chemistry, Snapdragon Chemistry, Erik Feldmann, PhD Technical Director, Early Stage Development and Testing of Cambrex, and Hannah McLachlan, PhD Project Scientist also with Cambrex. In this webinar, our team of experts will discuss technologies and case studies surrounding LPPS, peptide crystallization, method development for peptide impurity analysis, and much more. After their presentation, we'll be holding a live Q&A session. Feel free to submit questions as we go along. We'll get to as many as possible during the Q&A, and any that we can't get to, we'll follow up with post-event. Now, if you run into any issues, please try refreshing your browser first. If you need additional support, go ahead and submit a question via the Q&A, and I'll get you back on track. Finally, the webinar will be available for on-demand viewing. You'll receive a link via email as soon as it's available. Now, with all that said, I'm going to turn it over to Adrian to get us started.
Great. Thank you very much for the introduction. I'm happy to be able to kick off this three-part webinar to kind of talk through some of the approaches that we've been using at Cambrex to tackle peptide-based therapeutics. So before jumping into the peptides, just a brief introduction for Cambrex. We are a CDMO spread across 14 sites in the United States and Europe. And we cover a variety of services from very early stage development, IND-enabling services through analytical, solid-state chemistry, stability, storage, and testing, through drug substance services, so GMP and non-GMP manufacturing, from early stage to late stage and commercial process development, analytical R&D and validation. And in particular, we put a large focus in enabling technologies and specialized technologies such as being able to work with high potency and controlled substances, continuous flow R&D and manufacturing, particularly relevant for this webinar, complex synthetics and peptides, as well as biocatalysis development and manufacturing. So as the first part of this three-part webinar, I will be discussing at a relatively high level the approaches that we've been using at Cambrex to implement liquid phase peptide synthesis to develop more scalable approaches for peptides. So as many attending this webinar will be well aware, the demand for peptides has been growing quite a lot over the last 10 to 20 years, and it's become a major modality for drug discovery, with demands now in the order of metric tons. But still, SPPS still plays one of the major roles for supporting this manufacturing, despite the fact that this technology was really developed for discovery and lower kilogram scale manufacturing. And considering the basis for this technology, there's a large environmental impact due to the amount of waste that's generated. So there's a lot of room for positive impact through innovation in this space for more sustainable technologies. So at Cambrex, we've been putting a lot of emphasis on developing workflows for liquid phase peptide synthesis. And overall, liquid phase peptide synthesis is not too different from solid phase peptide synthesis. The main difference is removal of use of a insoluble resin that's used for solid phase peptide synthesis. Now, we recognize that for early applications and lower volume deliveries, SPPS is still really the technology of choice due to the much more limited development time compared to LPPS that's required before scale-up. But there's a lot of advantages that LPPS can offer over solid phase based approaches. So one of these is the use of more standardized batch reactor technology. So there's greater available global capacity at CMOs internationally. Since this is being performed in the liquid phase, you'll often see better reaction kinetics and coupling efficiency. And there's also a lot of advantages into process monitoring, where you can use very standard sampling and HPLC analysis to understand the progress of the chemistry without any needed sample derivatization. And one of the biggest impacts is improved volumetric efficiency, where you can see drastic reductions in PMI when using LPPS compared to SPPS. And this is shown well here in this plot taken from a paper published earlier this year comparing the average PMI for various stages of LPPS processes compared to SPPS, where you're often seeing drastic differences, sometimes within the order of magnitude, for the PMI for a liquid phase process compared to a solid phase peptide synthesis process. So at Cambrex, we've largely been focusing on the use of LPPS for the synthesis of shorter peptide oligomers, so usually in the range of 10 to 12 residues, where the plan is to then take these fragments and couple them together in downstream convergent synthesis of these fragments to make larger peptides, as shown graphically down here. And this is an approach that's also utilized with SPPS, where shorter fragments are made with solid phase and then coupled together in solution phase later on. But the same approach can be taken with LPPS. And liquid phase offers a nice additional flexibility on the protecting group strategy, where you can mix and match even within a given sequence or between different fragments for what types of protecting groups on your N-terminus you might be using. So a lot of our focus has been on the use of smaller C8 to C10 carbon esters that will solubilize these shorter oligomers, developing approaches for liquid phase synthesis to tackle byproducts from deprotections and coupling, use of more process-friendly solvents, and where appropriate, using combinations of flow and batch approaches for the synthesis. And so just to show this a bit more graphically for really the similarities between these different processes, solid phase and liquid phase has a lot in common. With solid phase, you're generally linking your growing peptide chain on the C-terminus to an insoluble cross-linked resin, where you then go through the coupling cycle of deprotection, washing of your resin with extra solvent, amide coupling, and then again washing and repeating back at the beginning of this cycle. So liquid phase is basically the same idea, but in this case you have a soluble hydrophobic tag, usually on your C-terminus, and then you can go through the same cycle. And phase is a great example of an approach that was used for this, using hydrophobic tags that are generally in the 1,000 grams per mole or greater molecular weight. And this allows for synthesis of very long peptides in the liquid phase. So we've been focusing largely on these shorter fragments, which allows us to be more in the 150 grams per mole range for molecular weight of the hydrophobic tag. And we've often also put a lot of effort into the use of Cbz technology, which offers a lot of advantages in terms of the workup that has to be done after N-terminal deprotection, where washing is not really required because the byproducts after Cbz deprotection will not interfere with the downstream chemistry. So we take a relatively holistic approach when we're getting into LPPS process development. We have a few key considerations that we're looking at. So one is the fragment identity. This is essentially your redshift and synthetic analysis of your full-length peptide, where you're looking for appropriate solubility as well as efficiency of your fragment coupling downstream. The tag selection, which is going to indicate what types of tag cleavage conditions you're going to use, as well as the tag molecular weight, which will usually depend on how soluble your individual fragment is. If it's a particularly insoluble fragment, you might need a larger molecular weight tag, or if it's a well-solubilized fragment, you can get away with these very small tags in the 150 grams per mole range. Another consideration is the protecting group chemistry, and I'm specifically speaking about the N-terminal protecting group here, where we consider what types of amino acids and sequences are present. So Fmoc chemistry requires basic conditions. So if you have sequences that are prone to diketopiperazine or other base-mediated processes, you need to avoid that. Whereas Cbz chemistry can sometimes not be compatible with certain side chains or tags. So to dive a little bit deeper into each of these categories, so looking at fragment selection, one of the key aspects here is understanding the solubility of proposed fragments for your full-length peptide. One of the first things that we'll do often is a paper exercise looking at the aggregation potential of individual proposed fragments. And there's a lot of tools that have been developed around knowledge from solid phase synthesis to understand the likelihood of aggregation for an individual fragment. And this can also provide insight into whether downstream fragment coupling may be difficult. Another thing we take into consider is our choice of C-terminal residue for each of these fragments. You want to avoid C-terminal residues that are prone to epimerization, such as histidine or cysteine or similar residues, and try to choose wisely around where you're going to be slicing up your full-length peptide into individual fragments. So looking at tag selection, as I mentioned previously, particularly insoluble peptide fragments will generally require tags that are more in the 500 to 1,000 grams per mole range, whereas more soluble fragments can often be at very low molecular weight tags. And the tag structure will also determine the cleavage chemistry once you get to the end of synthesizing your fragment. So while all of these different tags be cleaved generally under basic conditions, some of them also have other options that might be a bit milder. So just generic aliphatic esters, these are going to require base-mediated saponification in most cases, whereas benzylic and allylic esters can often be removed through hydrogenation, and the allylic esters can also be removed through palladium allyl chemistry. And then another category, similar to benzylic esters, 2,4-dialkoxybenzylic esters can often be removed with relatively dilute acid without deprotecting your fragment's side chain protecting groups. So we've put a lot of effort into developing workflows to utilize solid phase peptide synthesis to facilitate the transition into liquid phase. And we can use SPPS to synthesize proposed fragments, as shown in the cartoon at the bottom of this slide, and then actually install a variety of different tags to these proposed fragments, make sure they have appropriate solubility, but then also make sure that we're able to remove the tag at the end of the synthesis without decomposing or epimerizing various residues in the fragment. Then the final check that we'll do is make sure that we have appropriate kinetics and minimal epimerization of C-terminal amino acids when we do downstream coupling with other fragments. So this puts us in a good place to not waste a lot of time getting all the way through the end of an LPPS campaign and then realizing either we can't remove the tag or coupling of the fragments is problematic. So looking at the specific N-terminal protecting groups that we might want to use, there's a few things to consider and advantages and disadvantages for different approaches. So Cbz protecting group strategies have a preference for base-sensitive side chains or sequences, particularly sequences that are prone to diketopiperazine or aspartamide formation. And Cbz protecting groups also provide a very simplified workup, which can save you a lot of time and waste when you're doing the N-terminal deprotection. Fmoc protecting group strategy is preferred for side chains that are sensitive to hydrogenation conditions or that might inhibit hydrogenation, such as sulfur-containing residues can often interfere with palladium catalysis. And Fmoc protecting groups also have an advantage of much more rapid deprotection kinetics. And finally, another advantage for LPPS is the ability to mix and match with relative ease the types of protecting groups, even within a given fragment synthesis. For example, if you have a dimer or trimer that's prone to decompose under basic conditions, you can go through the initial portion of the sequence up to a certain residue with Cbz protecting groups. And then once you get to a residue that might not tolerate hydrogenation conditions, you can then switch to Fmoc protecting groups or other types of protecting groups with relative ease without having to switch types of equipment or make major changes to any sort of processing. So looking specifically at Cbz protecting group, or what we call here at Snapdragon Z-Flow, I've mentioned some of the general advantages of Cbz protecting group strategies, but also just looking at the types of hydrophobic tags that can be used. Since this is using palladium catalysis and hydrogenation conditions, you're generally limited to things like branched aliphatic esters, as benzylic esters will likely be cleaved under those conditions. But overall, the synthetic sequence is very similar to what you would see for most peptide syntheses, where you have an initial esterification, where you install your tag, and then you deprotect your N-terminal residue and enter the repeating cycle of amide coupling, deprotection, and then repeat to build out your fragment. And then once you get to your final full-length fragment, you can then cleave off the tag and isolate that as a stable building block to then be coupled downstream at a later time. And so we've utilized a variety of approaches for optimizing the N-terminal deprotection. One approach that's been quite successful for us is algorithmic optimization of the hydrogenation process. This is particularly useful in a packed bed reactor, where we can link this in with our LabOS software, where we can do online data collection using a UPLC for unattended and fully automated optimization of the process, exploring the design space around temperature, pressure, hydrogen equivalence, and residence time. And usually within the first 1 to 2 rounds, we end up at our fully optimized conditions that provides good throughput and rapid deprotection of the N-terminal protecting group. Now, when there's equipment limitations or we just want a general batch process, we have demonstrated this on scale with atmospheric pressure hydrogenation, and we found that this is efficient enough for our purposes, where within a few hours you're able to reach high conversions just by sparging under atmospheric pressure a batch reactor with a palladium on alumina catalyst. So this Z-flow approach has been demonstrated on multiple kilogram scale, up to an 8 mer so far in our labs, producing peptide fragments in very high purity after isolation. And this campaign that I'm showing in these pictures here was carried out completely in batch reactors, where we telescoped through the entire process and only isolated after tag cleavage at the very end, providing this peptide in very high purity, greater than 97 LC area percent, without any preparative chromatography being required. So there's sorry about the text and some of the graphical issues here, but we've compared the overall process mass intensity for this process, shown at the bottom left of this graph on this slide, and it measures up quite favorably compared to both existing LPPS processes and in particular compared to SPPS processes, where you're seeing over an order of magnitude improvement in the process mass intensity, where the main source of waste here is aqueous waste from the aqueous workups throughout the process. So looking more closely at the Fmoc protecting group approach, SPPS has an advantage where it's easy to remove the byproducts from Fmoc deprotection, namely dibenzofulvene, as well as the base, typically either piperidine or DBU or similar bases, just by washing the resin. You have to be a little bit more creative with LPPS, where you need something that can remove the highly hydrophobic dibenzofulvene byproduct. In this case, we utilize sulfonated thiol salt scavengers to remove around 97% of DBF each cycle. So in these chromatograms, you can see the formation of a DBF adduct. And then after just a simple water wash, you can see that that DBF adduct is fully purged from the reaction mixture, and we can move forward to the next step. So this process has also been demonstrated on multiple kilogram scale, out to an 11 mer so far in our labs, producing peptide in good purity. In this case, it was 95% LC area percent after hard cleavage of the still tagged peptide chain. And again, this also measured up quite favorably as far as the process metrics in terms of the waste being generated, where we're seeing about 94, a PMI of about 94 per amino acid compared to around 1,500 per amino acid for an average SPPS process. And again, the main source of waste here is aqueous waste. So looking forward towards future directions and investments across Cambrex moving into the space, again, we recognize that for early stage and small volume products, SPPS is still the leading approach, and it makes a lot of sense for these early deliveries, as there's much reduced process development compared to LPPS, and you can use turnkey, readily available SPPS reactor systems to do this, and then follow the typical pathway of large-scale reverse phase HPLC and lyophilization. But we've put a lot of focus into how we can use SPPS to lower the barrier to LPPS, using the strategies that I've mentioned on previous slides for fragment selection to make sure that we have the solubility that we need and can do downstream coupling as needed, as well as facilitating for tag selection to make sure that the tagged peptides have good solubility and that we can cleave those tags. And once we've got the fragment and tag locked in, then we can initiate the full-scale LPPS process development. Another piece of technology that we've been implementing is a fully automated lab scale LPPS system that can tolerate or accommodate both Fmoc and Cbz protecting group approaches. And this allows for unattended and fully automated synthesis of peptides using liquid phase, where you have an activation reactor, and for activating of your incoming amino acid, this can then be transferred to your coupling reactor. And after coupling and workup is complete, this can then be transitioned to a hydrogenation reactor, or Fmoc deprotection can be carried out in the same reactor and then repeat as we go through this process. With full control with our LabOS automation software and feedback control being enabled through integrated process analytical technologies. And apart from just general LPPS approaches, we've been investing in implementing relatively standard supporting technologies for peptide synthesis. This includes PrEP HPLC, tangential flow filtration with nanofiltration and ultrafiltration membranes for buffer exchanges and impurity purge, ion chromatography, as well as intermediate isolation technologies, including centrifugal decanters and filter dryers, and then standard API isolation approaches like lyophilization and spray drying, and then packaging under ISO 7 compatible conditions. And so to summarize my portion of this webinar, we've just discussed two main strategies, Z-flow or Cbz protecting groups and Fmoc LPPS. Both of these have been demonstrated in our labs on multiple kilogram scales at this stage, generating peptides in good to high purities. And we're starting to transition to demonstrate this on greater than 10 kg scale and further in the coming months. And so Z-flow is amenable to both batch and flow processing, flow in particular for the hydrogenation chemistry, whereas Fmoc is generally carried out in a batch process. And across the Cambrex network, we're able to scale this to 2,000 to 16,000 liter reactors for later stage processes that need to be further scaled up. But despite the capabilities within Cambrex, we are also willing to assist clients with transfer to third-party CMOs as needed without concern around IP restrictions. So with that, I'll conclude my section of this talk, and I'll pass the baton over to Hannah, where she can discuss some of the peptide crystallization.
Okay, thank you very much, Adrian. So yes, I'm going to talk about our crystallization screening and some basic initial crystallization process development that we carry out at Cambrex Edinburgh. So, as I'm sure you know, peptide crystallization screening aims to identify conditions which will result in a crystalline and developable solid form of a peptide. Quite often, peptides are initially developed as an amorphous material and also as salts, and these are quite often very fluffy and very hard to handle. Peptide crystallization screening is often complex, with a narrow crystallization operating range compared to other less flexible small molecules, and this also leads to a high risk of gelling, aggregation, and fibrillation with the peptides. Crystalline solid forms offer many advantages over amorphous forms, including improved stability and better physical and chemical properties. As these improved properties can lead to processability improvements and also therefore improved handling for further development. And if you can get a crystalline form, you can also then quite often get full structural determination through single crystal analysis, which is very helpful. We have also found that crystallization processes can be used as purification techniques, and this can lead to significant cost savings overall when compared with chromatography preparation techniques. So I'm now just going to give an overview of how we would tackle crystallization screening, and then we'll also have some examples towards the end of my section. So what we aim to look for or cover in our crystallization screening projects is we normally start with solubility determination and a selection of aqueous and organic solvents and buffer systems to determine potential crystallization media, and this also then acts as an initial crystallization screen. We would also look at the buffer capacity of the material, which is what concentration of buffer is required to maintain the pH at a certain set peptide concentration. Any buffer selection we do is always carried out in conjunction with any clients, and this is utilising the internal Cambrex expertise and client knowledge of material to ensure that the most appropriate systems are selected and that we do not cause any damage to the peptides that we know would happen. Our first technique we look for is crystallization screening of the peptide using multi-well research plates. This allows us to use a very small amount of peptide material per experiment and screen a large number of crystallization conditions at once. At our site, we have over 100 buffer-based systems available in-house, as well as other techniques using just organic solvents or aqueous media if required. Initial indications of crystallinity of material from these these systems can usually be obtained within 2 weeks. We will also look at more conventional organic and aqueous screening experiments, which utilise techniques such as maturation, anti-solvent addition, slow cooling, evaporation, just to give as much chance as we can to prepare a crystalline material. In addition to this, we would also can look at salt swap experiments through salt screening. So this can be done through both direct acid addition as well as resin salt swap techniques, just again to see whether any alternative salts can be found which may be more stable than a potentially previous prepared salt. So just to go into a little bit more detail about our research plates. So we use these, as mentioned, for initial peptide crystallization screening in both buffer and organic and aqueous solvent systems. And this system we have found offers many advantages. Generally, it uses small amounts of material per well. So this can be less than 1 milligram of peptide input per well. It just depends on solubility of the material. And we can screen up to 24 conditions. So this is different pHs, different anti-solvents, different buffer components, different salt additives, and things like that all at the one time. We can also store these plates at either ambient or, if the peptide is particularly unstable, at 5 degrees Celsius securely, so we can monitor for any differences in temperature effects. So how these work, mainly shown in the image at the bottom there on the left-hand side. A small amount of the peptide is dissolved in solution and then added to the post along with an equal volume of the crystallization reagent. The crystallization reagent itself is then added to the wells of the plate, and these are sealed. This just allows the system to be a saturated environment. And if there's any volatile components within the buffer or anti-solvent system, this will then also go through vapor diffusion. These experiments are monitored initially through microscopy to see for signs of differences in the posts. So the two images we have there, the middle one there is an oil we have seen form through over time, and then the image on the right-hand side there is showing little small rod-like crystals that formed of a specific sample. So once these potential crystallization conditions have been identified by PLM, and we will also use cross-polarized light to see whether they are biorefringent, we can then re-prepare them on a larger scale for further analysis. So we will quite often carry out various attempts to prepare them in different systems. So initially we will look for direct replication of vapor diffusion conditions on a larger scale. This is to allow us to get more material for analysis, but we will also look at concurrently slurry trials. So these will be carried out at various temperatures to offer potential alternative preparation routes that might be more scalable in the future. These are initially completed at the same concentrations and temperatures as were done in the research plates, and if successful, we will look at further peptide concentrations to test if we can identify other conditions, or if the gel where previously they have formed a nice solid, we will look at altering different things to try and see whether we can get other crystallization conditions identified. All material that we prepare will be analysed to determine the nature of the solid. This includes through our XRPD, so we have throughput XRPDs, UPLC for purity in any degradation, NMR to check for degradation in any solvent content, and also thermal analysis. We also have KF and GC if there's sufficient material. And this is allowing us to identify any improvements compared to the input material, so we can then focus on different areas we need to work on. So we have, in certain experiments and projects, been able to then go on to doing some crystallization development. So quite often these projects can carry on from our peptide screening work. So screening work can identify a crystalline form for further studies, with indications therefore of pH, excipients, antisolvents, target concentrations in any salts that are formed required to prepare our desired form. Our initial crystallization development program then aims to deliver a process which is scalable with regards to the inputs available and crystallization processes themselves. This then enables production of larger quantities of the target form for use in further studies, such as formulation assessments and potentially bioavailability studies. Isolation and drying studies are also often required to identify conditions which ensure the crystallinity is maintained, along with purity and other important crystal properties, while removing any excess solvent. And we can do this ourselves as well. So we have multiple different setups, and we also have a humid drying setup, which allows us to test different humidities for drying conditions if a hydrate was to be formed. So just a very quick overview of how our early stage peptide crystallization development typically works. So solubility, again, on metastable zone width assessments are carried out to identify potential solvent systems and limits for the range of crystallization conditions. We then move on to small-scale crystallization trials to identify potential pathways for the crystallization. So these can be done on a 20 milligram scale with overhead stirring. And then we would move on to crystallization development to define the process. So in-house, we have vessels that range in size from 50 milliliters up to 5 liters, so we can look at different conditions and scalability. So I'm now going to give a few examples of peptide crystallization projects we have worked on in the past and how we managed to get material. So our first peptide crystallization screening was an amorphous peptide salt, which was an acetate salt, and this was received for initial screening. We looked at the solubility of the material and found what it was soluble in. In this case, it was highly soluble in water, which allows us to then use this as the input from our research plates. We also looked at buffer capacity and then a total of 168 research plate sitting drop trials. From these, potential crystalline hits were observed in multiple conditions, as we can see there from the top image. That is again an image from a research plate looked at for PLM. We then managed to complete small-scale trials to test initial crystallization conditions, which were indicated by the research plate conditions, and prepare material for further analysis. Crystalline solids were recovered. However, this material that was prepared was found to be unstable under storage at even at minus 20 degrees Celsius. We then looked at different alternative systems. So in this case, we looked at salt swaps. So we looked at a hydrochloric and a sulfate salt. These were the forms targeted at the client's request. We successfully prepared a HCl salt and then used this for further sitting drop experiments. From these experiments, crystalline material was identified and recovered. And then this was again re-prepared on a larger scale for further testing. This HCl salt was found to be more stable. We ran out of material due to very limited material availability at this point. And the client was happy with our results, so they could move forward themselves. So moving on to a different project. So this was, in this case, it was an amorphous peptide hydrochloride salt, which again we received for peptide screening. We again initially used material to carry out solubility, buffer capacity, and sitting drop trials. From the sitting drop trials, we had limited crystalline hits. In this case, the material quite liked to gel in buffer-based systems. We then, from the solubility work, we had shown that we had two potential crystalline patterns, so pattern 1 and pattern 2. And we then looked at aqueous organic crystallization trials to test further solvent systems based around what we'd seen here. So these were water-based systems with potential antisolvents. And these used things like temperature cycling, antisolvent addition, slow cooling, and evaporation. From these techniques and trials, we obtained a further 3 crystalline patterns. And it was noted on drying at 20 degrees Celsius under vacuum that all solids formed a single consistent form, which was pattern 3. We looked to do a scale up and we successfully prepared material on a larger scale we successfully prepared the pattern 3 material on a larger scale via pattern 1. And this material was indicated to have likely be a hydrated form with low solvent content. Initially, the amorphous material was not particularly chemically stable at ambient and had to be stored at minus 20 degrees Celsius. After crystallization, material showed good physical and chemical stability. So it was stored for up to 1 month at stress conditions, which included storage at 40 degrees Celsius, 75% RH, and 25 degrees Celsius, 60% RH. And at both conditions, the purity stayed maintained and the form stayed constant. So we can see there from the impurity tables at the bottom, we have on the left-hand side, we have the purity of the crystalline material, which was the stability input. And on the right, we have the material after 4 weeks at 25 degrees C, 60% RH. And as we can see, the area percent purity is very consistent. This project then moved on to a crystallization process assessment. So following identification of the most stable crystalline form, which in this case was pattern 3, we looked to do initial crystallization assessment using approximately 20 grams of material received from the client. So initially, we carried out trials in our 20 ml vials with overhead stirring, which is the smaller image there. This was then followed by transfer to a 100 ml EasyMax system, where we used a blazemetric probe as PAT to monitor the process. So once the process was identified, we used a larger scale batch. We prepared a larger scale batch, and the crystallization material was then used for initial pre-formulation and bioavailability studies, which were also carried out at Cambrex, Edinburgh. So just to give an idea of sort of the trials we faced during these crystallization process assessment. So we noted quite long filtration times on a relatively small scale, so about 5 minutes on about a sort of 2.5 gram scale. We carried out looking at different cooling rates and anti-solvent addition rates, and we found that these improved crystallization control and particle size, which therefore reduced our filtration times. Related to our filtration times, we also noted potential gel formation during the filtration. This is just due to, again, the long time it was taking to filter through the filter media. We did discover that if we added a small nitrogen blanket to the filtration setup, in correlation with the improved particles, this then reduced this risk of gel formation, leading to a much more manageable material. Initially, we had some low isolated yields, so we looked at different final solvent ratios, temperatures and aging periods, and through these we managed to identify the optimal condition to achieve the maximum yield, very similar to what our theoretical yield looked like from HPLC analysis. Also, we did note some high residual solvent content. So we then, as mentioned, we looked at different drying conditions and things like, as mentioned, drying times, humidities, and even temperature in this case. We managed to identify suitable conditions to provide high purity solid with a low residual solvent content, so well within the ICH limits for the material. It was ethanol, and I believe our residual solvent content was about 500 ppm. While maintaining the appropriate water content to maintain form. This initial developed process has provided a route to isolate material with suitable crystal properties for further developmental studies. So my final examples here, this is a slightly different setup. So we were looking at first salt screening. So in this case, again, we received amorphous acetate salt, and client, particularly in this case, wanted to look for alternative salt systems because they knew that their acetate salt was not particularly good for the next steps in their system. So they asked us to look at both traditional salt screening processes and also using research plates as an alternative. So initially, small-scale trials were conducted using 8 counterions and adding either 1.1 or 2.2 equivalents of acid directly to the peptide, which was dissolved in water. This was dissolved at 20 degrees Celsius, and then precipitation occurred through either anti-solvent addition or cooling, or both. Crystalline solids were identified and recovered from HCl, methane sulfonic acid, and L-tartaric acid. On the XRBD stack there, we have the HCl and the methane sulfonic acid diffractograms, showing the different peaks. An NMR analysis of the salts indicated potential salts had been formed from these systems. I think in this case, CAD was also carried out on the hydrochloride salt and the sulfuric salt, just to make sure that we had what we thought we had. We did note on this particular exam, on both of these salts, that as we dried, we did lose some of the crystallinity. Experiments then were carried out using research plates through paired solutions with peptide and water with 0.5 molar acid stock solutions added to the both, along with the peptide and anti-solvent addition systems added to the wells. This was just looking to see, there was concerns potentially about degradation on direct addition of acid. So this was just an alternative system looking at a more dilute method to see whether we could get solids out. So after 1 day, precipitation was observed in 16 different experiments that were compared out, and the crystalline material was observed in phosphoric acid and 2-propanol water after 1 week. Oiling was also observed in one experiment, so we did note that we could get oiling if we had the conditions wrong. Again, this client was very happy with the results and came back to us when they had more material. So from this, we had 4 potential salt systems identified, as mentioned previously. So the HCl, the methane sulfonic acid, the L-tartaric, and the phosphoric acid from the plate system. The client asked us to look at initial trials to identify a salt rate procedure because they wished to have a free base form as well as an option. So this was carried out using the acetate salt input, and the procedure was identified, and this was successfully then carried out on a small scale, so about 500 milligrams. Then this was scaled up at client request to 1 gram, 5 gram, and 10 gram, and all were showing good good purity results and consistency in what was occurring in the system. Initial small-scale trials were then completed to attempt to prepare the crystalline salts. From this, HCl was found to have 4 different forms over the different trials, and the methane sulfonic acid and L-tartaric acid salts were also successfully formed, again, on a larger scale. The salt chosen to move forward was the BIS HCl salt, and this was selected by the client in conjunction with our experts at Cambrex to determine what was the best for what their needs were, and in terms of things like purity, stability, and all of these, and solvent contents, and all of these other factors. Following this, further solubility work was carried out in the target salt form to ensure that we knew as much as we could about the system before moving forward into any further scale-ups. This was then followed by metastable zone with determination in the preferred solvent systems. Initial crystallization development was then carried out, and this used propanol water-based systems, so both 1-propanol and 2-propanol were carried forward because they both looked quite favourable in terms of yields and purities. From both, crystalline material was recovered and maintained on drying, and a high solid purity was recovered. So we can see from the impurity tables there, the top one is the recovered solid material, and it's got about 98.2% area. And the input material, which is the bottom one there, is showing about 95.5% area purity. And we can see there's a reduction in the number of impurities within that peak table there. And also any of the purities that were retained were not growing at this time. So that is it from me for the moment. I will now pass on to Erik to discuss his section.
Thanks very much, Hannah. So yeah, I wanted to introduce at a very high level our solutions for method development, in particular for purposes of ID, assay, and impurities for peptides. And typically we use this approach, the same type of approach for small molecules, but we have a lot of experience utilizing this type of approach for peptides, not only for the API, but also for drug product matrices. And typically what we're going to use is what we call a rapid and data-rich method development through unbiased screening. And I'll get into some of these various ways that we can screen all of these parameters in the next couple of slides, and then I'll provide some case study examples to take us home. So typically what we're going to utilize in the analytical development laboratory is a matrix where we can evaluate the stationary phase column chemistry, the mobile phase chemistry that is the most suitable for the compound of interest. And so typically we're going to take a look at the structure of the compound, understand its unique chemistry, and then apply a smart matrix of stationary phases and mobile phases. And based on those that we select, we can then vary different parameters of the matrix, including column temperature, mobile phase flow rates with respect to pressures that are suitable for those columns, and then also mobile phase additives. So typically you're going to start with an acidic additive for your mobile phase, but not always, depending on the type of compound that you're trying to analyze. And then detection method. So in the analytical development space, we're pretty much always starting with a UV-based approach using a photodiode array detector, but based on the specific analyte, you can also couple that with another universal detector like charged aerosol detection, evaporative light scattering, Or if you're also looking for mass spec information, you can have a PDA detector followed by a mass spec detector in the back end to get that mass level information as well for impurities analysis. And also what we're going to leverage is force degradation studies to maximize the potential of that method's use. So what does this system look like in the Cambrex laboratory? So we have several systems that are outfitted very similarly to the one that you can see here in the diagram, where we have this particular instrument is the Waters Acuity H-class UPLC, which is our workhorse for analytical method development activities. And typically what we do is we outfit these with multi-column changers where you can screen 6 different stationary phase columns all in a single run, and then also add on the addition of solvent select valves where you can screen up to 9 different solvent combinations with those 6 different stationary phases. And then a sample manager that also has a thermostat control to where you can screen low temperature all the way up to high temperatures, in some cases as high as 60 to 80 degrees. And then what you also see on the right-hand side of this stack is the PDA detector, which would be the primary detector, followed by, in this case, PDA into a mass spec detector. In this case, this is a QDA system, but we also have multiple that have CADs or ELSDs depending on the use. So what does this workflow look like? Well, you work with the client to understand what type of a landscape they're looking to evaluate, and then also what the method's intended use is. If you're going to be utilizing this method for the API release and stability, that's one area where you define what type of a method you're looking for. If you need this for an impurities method, that can change the way you develop the method. But in the couple of case studies that I'm going to show you, we'll also utilize the same type of approach for peptide drug products as well. And so those are going to have an additional step where you may need to leverage extractions of the drug product matrix, but ultimately you can still take the same type of approach for drug product as you can for the API. So typically what you would start with would be a generic gradient for purposes of efficiency to put together a large screening data set. You'll start with a generic gradient of approximately 2 to 5% per minute of your organic phase. And then based on the chemistry of the analyte, you then select what are the most suitable starting conditions of the gradient, and then also which additives you may want to include in your mobile phases. If you have access to any intermediates or impurity markers, you can take those together with the API and create either a resolution mixture, or you can have them as separate samples that can be injected for the purposes of evaluating resolution while you're screening. And then utilizing this UPLC system, you can then screen up to 6 columns in a single run. And oftentimes you can screen 12 or 24 or even more columns across a given data set. You'll put together your force degradation samples in parallel while you're designing these experiments, and then screen all of those across the most appropriate force degradation conditions, whether it would be photolytic decay, thermal analysis, acid-base hydrolysis, or oxidative stress. And then the mobile phase selections, typically you're going to use the classic solvent selectivity triangle where you would rely on either an ACN gradient, acetonitrile, methanol, or THF. And oftentimes, depending on the chemistry of the analyte, you may also include blends of those 3. Once you perform that initial primary screen, you'll down select your lead 1 to 2 columns. And then based on the selection of those 2 columns, you can then get into even more tightly refined tuning of the gradient ramp itself, temperatures, flow rates, and then additive percentages or concentrations in the mobile phase. Once you've identified that final lead column and preliminary conditions, you can then do your final optimization and then pre-validate the method for its to make ready for either early phase qualification activities or late phase validation activities. So the first example that I wanted to show is a drug product containing a peptide that was in a solid implant, and the customer came to us with an initial method that they needed high resolution to maximize impurities analysis. So in this case, the API in the drug product was cyclosporine A, and we were able to identify or source several commercially available cyclosporine variants and what you see here in this resolution mixture. And you can use that resolution mixture of all these different cyclosporine variants as a way of performing your initial screening activities for the method to improve its resolution before you get to force degradation studies. So if you take your resolution mix, you can use that as the initial sample, screen through the initial columns that you'd like to screen, depending on how many columns the customer wants to screen. You have that conversation together, and then evaluate based on the number of columns, how many different mobile phase conditions, temperature conditions, and subsequent screening activities afterward. In this case, if you're looking to keep a method that's suitable for mass spec, that will also define what additives you use. If you want it to be mass spec friendly, you use mass spec friendly additives. If you're looking to maximize sensitivity by UV, oftentimes you may go the route of using phosphoric acid as an additive. And then of course, you can do peak purity analysis as well using the photodiode array detector as you're trying to understand your impurity profile. And if you have a mass spec detector coupled in line with the PDA, you can also make an assessment of what those impurities are along the way as you're developing the method. So just as an early example of how we would take a customer's method, in this case, they came to us with an isocratic method that had very low resolution on the left-hand side. You can then take a simple acetonitrile gradient, and then depending on the columns that you're choosing, you then can go up, increase the column temperature. In this case, we show from 40 to 60 to 80 degrees Celsius with a simple ACN gradient, and you get dramatic improvements in peak shape compared to that what you see on the left-hand side at 40 degrees with a simple isocratic gradient. And for this same customer, we then took it a step further when we started looking at force degradation analyses. In this case, on the left-hand side, you can see where we're screening 7 different columns or stationary phases, all of the same sample, and you can see dramatic changes in the impurity profile. So at the top, you see approximately 33 impurity peaks, down to 27 impurity peaks at the second from the bottom, and then as high as 41 and 40 impurity peaks, depending on the stationary phase that you choose. So based on that initial assessment of columns, you can then down select 1 to 2, move over to the right-hand panel, then you can take that lead column, 1 or 2 columns, and then screen things like temperature, flow rate, gradient ramp rate, and then the mobile phase strength. So in this case, varying the percentages of the gradient or even the composition of the organic to maximize your resolution for an impurities method. So the second case study and final case study that I'm going to showcase is one for a drug product where there was a cyclic peptide coupled to a 30 kilodalton PEG. And in this case, the customer came to us with a request for multiple methods. They wanted a method for the API to understand the impurity profile of that of the full conjugate, as well as the cyclic peptide intermediate to understand what impurities were forming. So it needed to be mass spec appropriate. And then also a method for free PEG that hadn't been coupled in the final bioconjugate. And then also a sizing analysis type method to understand what the size profile is of the final bioconjugate. So what I'm showcasing here are 4 of the 4 methods that we developed for them. In the initial top left-hand panel, you'll see our approach for mixed mode column screening to try and get a method to separate out a massive impurity that was observed under the main peak. So if you look in the top left panel, the green trace, you don't see an impurity underneath that main. But as we started to screen through different mixed mode columns, you could see a marked shoulder in the pink trace, which in this case was approximately 20% of their material. And the customer didn't know about this until we were able to resolve it using this type of approach with mixed mode column screening. So if you go one panel to the right, this was the final method that we developed for them so that they could actually resolve that impurity away and then move that method over to scale up for impurity isolation and impurity identification purposes. So we took that method then, scaled it up for a semi-prep column, isolated that impurity, and then collected high-res mass spec information and NMR information to ID what that species was. In the lower left panel, what you're seeing is a method that the customer initially came to us with in the black trace to resolve impurities in their peptide intermediate. And then we optimized that method on the blue trace to separate out as many impurity peaks as possible. And it had it mass spec friendly so that we could collect as much mass spec information as possible to understand what impurities were forming. That's the blue trace. And then the final method that we developed for them was a size analysis method for the full bioconjugate. And what you're seeing here is size exclusion using refractive index chromatography that leverages a calibration curve using known PEG molecular weight standards to determine the molecular weight of their full bioconjugate. And what we've invested in at Cambrex is now taking this a step further, going from refractive index detection to then utilizing a light scattering detector where you can run SEC with moles detection in the back end, so you don't have to rely on a calibration curve using something, in this case, that may not be the most suitable standard for your analyte of interest. Because in this case, it's a peptide coupled to a 30 kilodalton PEG, not a free PEG itself. So to summarize, some of the other requests that we're getting recently is also trying to move the final method readiness over to commercialization, making sure that the method is robust and sound to support routine QC use for late phase customers. And one of the requests that we're seeing is that for the purposes of ID of that peptide, where we take advantage of our high-res mass spec detectors, where we sequence that peptide, or in this case, if it's a large polypeptide like a protein, we may collect sequencing information to ID that material, and we'll summarize all of that information in the development report and not necessarily include it in the qualification or validation studies. Same thing with robustness studies. Quite often now we're hearing customers would like to evaluate the method robustness as part of development, and then that can be referred back to and summarized in the qualification itself and not repeating it in the actual qualification study itself. But lastly, just one thing I'd like to end on is that quite often when we get to late phase use, customers are looking for a simple and robust method to support that routine commercial QC. So it's important and very valuable to conduct these robustness studies as part of the development so that you don't end up in routine QC where you're having system suitability failures because the robustness of the method hasn't been evaluated. Okay. And with that, I'd like to conclude and open it up to Q&A.
Okay, thank you so much, Erik. I'm going to pop back on and just, thanks to all of our speakers for that great presentation. We'll get right to our Q&A. We'll start with our first question. And that question is asking, if possible, can you please comment on the general impurities typically observed in LPPS and SPPS? Who can help us with that one?
I can tackle some of that. Yeah, there's a lot of impurities that can be formed due to side reactions and peptide synthesis. In between LPPS and SPPS, it's a lot of very similar impurities for the most part. So there's the ability for side chain deprotection during the synthesis, Cambrerization of both your C-terminus as well as incoming amino acids are often observed to some degree. Incomplete coupling or double coupling is the main impurity. So you'll see deletions in your sequence or double inclusions. Let's see. N-terminal capping is another very common impurity that you'll see. This can be both from the dibenzofulvene. So this is more of a problem with liquid phase actually than it is with solid phase. If you don't purge the DBF well enough, that can actually react directly with your N-terminus for irreversible capping. As well as if you're using uranium-based coupling reagents such as TBTU or HA2, those can cap your N-terminal as well. And then there's impurities related to, in liquid phase in particular, tag cleavage, where you'll actually cleave off of your hydrophobic tag. This can be observed with solid phase, but usually you'll just wash away the cleaved peptide, so you might not observe it in your LC. So a lot of things can happen. Most of them are very similar between solid phase and liquid phase.
Okay, very good. Thank you. I think we have time for one more question. And I think this question is for Hannah. It's asking, does Cambrex help clients to identify which form will have favorable bioavailability? What do you think, Hannah?
So we can definitely look at multiple different forms and things like purity and all different standard things that we would also look at for small molecules. In terms of bioavailability, we can do some initial trials, at least at Cambrex Edinburgh, and we also have other partners that we can work with, or if the client has any partners, we're happy to work with them. In terms of specifically for bioavailability, this is not my area of expertise, but we do have people who could talk to you about this. So if you had any questions about that and wanted to get more information, please reach out to us separately and we'll be happy to contact you and to talk with you about our potential options for future work.
Okay, thank you so much. And that is our time. If we did not get to your question, no worries. We will be sending these to our presenters and they'll be answering them post-event. You'll receive a link via email after this is available to see this again on demand. Thanks for watching. Thanks to our presenters so much. And thanks to you, the audience. We'll see you next time on Outsourced Pharma. Have a great day.